In 1888, a British researcher named Frank Wilcox documented something that would quietly reshape how we understand plant growth. He was studying root nodules on legume plants in rotational farming systems across England, and what he found wasn't just interesting, it was economically impossible to ignore. These tiny bacterial colonies were pulling nitrogen directly from the air and converting it into plant-available nutrients at rates that matched or exceeded what farmers were buying in bags. The discovery confirmed what agricultural societies had practiced for millennia. Soil wasn't just dirt, it was a living factory that manufactured its own fertility when left undisturbed. For decades after Wilcox's work, research institutions across Europe and North America expanded on this knowledge, documenting how microbial communities could replace external inputs almost entirely. But then, in the span of about 20 years, this entire body of knowledge was systematically deprioritized, buried under mountains of research funding that flowed in exactly one direction. A detail in that story was deliberately erased. By the 1950s, agricultural extension services were publishing standard recommendations for commercial farming. The lists were straightforward: tillage depth, planting density, irrigation schedules, and input applications. Every category had its place. Soil management appeared where you'd expect it, tucked between crop rotation and pest control. But there was something strange about how one particular practice was classified. Biological soil management, the systematic cultivation of microbial communities to generate fertility, wasn't listed under soil management at all. It appeared in a completely different section, one reserved for experimental techniques and unproven methods, right alongside untested theories and fringe practices. This wasn't an oversight or a formatting error. The reclassification was deliberate, and it happened at exactly the moment when a competing technology needed the market cleared. That practice was living soil, the self-regenerating fertility system. And for thousands of years before industrial agriculture existed, it was the only system that sustained continuous cultivation without external inputs. From the terra preta of the Amazon to the centuries-old rice paddies of Asia, every major agricultural civilization built their food security on one fundamental principle: feed the soil life, and the soil life feeds the plants. Living soil wasn't a technique, it was the foundation of agriculture itself, the biological engine that powered crop production long before anyone conceived of mining nitrogen from the atmosphere or extracting phosphorus from ancient seabeds. It worked not by forcing growth through chemical intervention, but by creating conditions where trillions of microorganisms performed the same nutrient cycling that sustains every natural ecosystem on Earth. And for most of human agricultural history, there was no alternative, because there didn't need to be one. Estruturou argumentação com evidências históricas e escala global. Preciso estabelecer a importância histórica com fontes antigas e evidências de escala. Vou usar exemplos de civilizações antigas, documentação de rendimentos e evidências de que isso era o padrão global. The earliest agricultural texts don't mention living soil because they didn't need to. It was assumed. Chinese agricultural manuals from the 6th century BCE describe elaborate composting systems and microbial inoculation techniques without ever explaining why, because every farmer already understood that soil health determined harvest size. Roman agronomists like Columella wrote extensively about maintaining soil life through crop rotation and organic amendments, documenting yields that rival modern organic systems. Medieval European monasteries maintained productive gardens for centuries on the same plots without any external fertilizer, relying entirely on managed microbial activity and nutrient cycling. Indigenous agricultural systems across the Americas, Africa, and Asia all operated on identical principles: maintain the soil ecosystem, and fertility maintains itself. The terra preta soils of the Amazon, created through deliberate microbial management and biochar addition, remain more fertile than surrounding rainforest soils 1,000 years after they were last cultivated. This wasn't primitive farming or low-yield subsistence agriculture. These were sophisticated biological systems that sustained populations for generations without depleting the resource base, and they did it by treating soil as a living community rather than an inert growing medium. And then, beginning in the 1940s, living soil disappeared from mainstream American agriculture. Not slowly, not through gradual replacement as farmers tested alternatives, but systematically, as extension services, research institutions, and agricultural colleges shifted their focus almost overnight. It wasn't because the biological approach stopped working. Farms using living soil methods in the 1950s were still producing competitive yields with virtually zero input costs. It wasn't because of disease or pest pressure that biological systems couldn't handle. And it wasn't because farmers rejected the approach. The shift happened from the top down, driven by research funding, university priorities, and extension service recommendations that suddenly, uniformly, began promoting a completely different model. Within a single generation, the biological approach that had sustained agriculture for millennia was reclassified from standard practice to alternative method, and then from alternative to outdated. The knowledge didn't vanish entirely. It was archived, moved to the back sections of agricultural libraries, and gradually excluded from mainstream agronomic education. This isn't a story about farming techniques. It's a story about economic architecture, about how an industry built on annual sales couldn't coexist with a system that generates its own inputs indefinitely, because living soil has one characteristic that makes it fundamentally incompatible with the modern agricultural input market. Once established, it doesn't require repurchasing. A healthy soil microbiome regenerates itself season after season, year after year, performing the same nutrient transformation services without needing replacement or supplementation. There's no subscription model, no recurring revenue stream, no annual purchase cycle. And in the 1940s, as chemical companies transitioned from wartime production to peacetime markets, they needed agriculture to operate very differently than it had for the previous 10,000 years. They needed farmers to buy products every single season. Living soil, by its very nature, eliminated that need. So the system that sustained human agriculture since the Neolithic had to be replaced, not because it failed, but because it succeeded too well at making farms independent. The file opens in 2011 when a USDA research team published findings that would quietly validate what traditional farmers had known for millennia. They weren't studying ancient techniques or organic methods. They were documenting next-generation biofertilizers developed by companies like Pivot Bio and Join Bio, corporations engineering microbial strains to fix nitrogen directly on corn and wheat roots. The results were startling. Test plots using these biological systems reduced synthetic nitrogen application by 25 to 50%, while maintaining or increasing yields. But buried in the discussion section was a sentence that reframed the entire conversation about soil fertility. The researchers noted that these engineered microbes were performing the same functions that naturally occurring soil communities had been doing in undisturbed agricultural systems for centuries, and that the primary innovation wasn't discovering new biological mechanisms, but rather reintroducing biological capacity that conventional farming had systematically eliminated. The problem wasn't that living soil was inefficient. The problem was that it had been so thoroughly removed from agricultural systems that even its basic functions now required re-engineering and reintroduction. 8. Demonstração técnica do produto. Living soil operates through an interconnected web of microbial processes that collectively replace most external fertilizer inputs. Nitrogen-fixing bacteria, primarily rhizobia, but also free-living species like Azospirillum and Azotobacter, colonize root zones and convert atmospheric nitrogen into plant-available ammonia through enzymatic processes powered by photosynthesis-derived sugars from the plant. Mycorrhizal fungi extend root systems by factors of 10 to 100, accessing phosphorus and micronutrients far beyond what root hairs can reach, then trading those nutrients for carbon compounds the plant produces. Phosphate-solubilizing bacteria secrete organic acids that break down insoluble mineral phosphates in soil, making them bioavailable without requiring mined rock phosphate or synthetic processing. Decomposer communities of bacteria, fungi, and protozoa break down organic matter into humic compounds that improve water retention, cation exchange capacity, and nutrient holding ability. The system is self-regulating. Populations of beneficial microbes suppress pathogenic species through competition and antibiotic production, reducing disease pressure without chemical intervention. And unlike fertilizer applications that deliver nutrients in a single pulse, these biological processes operate continuously throughout the growing season, matching nutrient release to plant demand in real time. A well-developed living soil system can generate 100 to 200 pounds of plant-available nitrogen per acre per year, solubilize enough phosphorus to meet crop needs entirely, and maintain optimal micronutrient availability, all without a single bag of purchased fertilizer. While living soil was being systematically sidelined, synthetic fertilizers were being engineered for a very different set of priorities, not for long-term soil health or biological sustainability, but for immediate measurable response, uniform application, and predictable supply chain management. Nitrogen fertilizers were formulated for maximum water solubility, so they'd show visible results within days of application, which also meant they'd leach rapidly through soil and require reapplication. Phosphorus fertilizers were processed into forms that plants could access immediately, bypassing the natural slow-release mechanisms that mycorrhizal fungi provide, which created dependency on continued applications as soil biology degraded. Potassium, micronutrients, and soil amendments were all optimized for the same characteristic: fast, visible response that could be measured in a single season and would need to be purchased again next year. The formulations weren't designed for soil health or long-term fertility. They were designed for market efficiency, consistent NPK ratios that could be manufactured at scale, shipped globally, and applied with mechanical precision. The entire system was built around one unstated premise: soil is an inert substrate that holds plants upright while you feed them through chemical injection. And once that premise became the foundation of agricultural education, research funding, and extension service recommendations, any system that contradicted it, no matter how proven or effective, became definitionally obsolete. Identified standardization barriers that systematically excluded living soil adoption. Aqui preciso mostrar como a padronização criou regras invisíveis que excluíram living soil. A indústria de fertilizantes criou padrões que favoreciam produtos químicos. The modern agricultural system operates on standardization that appears neutral but functionally excludes biological approaches. Crop insurance requires documented fertilizer applications at industry standard rates, which means farmers using living soil methods may not qualify for coverage because their input records don't match. Actuarial tables built around chemical systems. Agricultural credit, the loans that finance planting and operations, often mandates specific input purchases as collateral protection, ensuring that biological fertility methods, which reduce purchased inputs, also reduce access to capital. University extension services publish fertilizer recommendations based on soil tests that measure only chemical availability, completely ignoring biological capacity, which means a soil with thriving microbial communities fixing nitrogen and solubilizing phosphorus will still test as deficient and trigger recommendations for synthetic applications. Certification systems, equipment design, seasonal calendars, and even the physical infrastructure of modern farming, from fertilizer spreaders to application timing, were all built around chemical inputs. Living soil doesn't cooperate with any of it. Microbial communities don't operate on application schedules. They function continuously based on temperature, moisture, and plant demand. You can't spread them with a spinner spreader or inject them with a knife applicator. They don't come in standardized NPK ratios that fit existing recommendation systems. The entire architecture of modern agriculture was designed around products that could be sold annually, and any system that generated its own fertility became, by definition, non-standard. But there's a deeper structural problem that explains why living soil had to be replaced, and it has nothing to do with farming. Living soil doesn't require repurchasing. Once you establish a healthy soil microbiome, it regenerates itself indefinitely. You plant a cover crop, the roots exude sugars that feed bacterial populations. Those bacteria fix nitrogen and solubilize phosphorus. The crop grows, residues decompose, and the cycle continues season after season without any external input purchase. Compare that to the synthetic system. You apply nitrogen fertilizer. The crop uses what it can. The excess leaches away. Soil microbial populations that would have fixed nitrogen naturally are suppressed by the chemical nitrogen they're suddenly swimming in. And next season you need to buy nitrogen again because the biological capacity to generate it has been eliminated. You apply phosphorus, it binds to soil particles, becomes unavailable. Mycorrhizal fungi that would have accessed it can't compete with the chemical pulse. Their populations decline. And next year you need to buy phosphorus again because the biological system that would have solubilized it is gone. The synthetic system doesn't just feed plants, it actively dismantles the biological systems that could do the same job for free. And once those systems are gone, once soil microbial communities have been degraded through decades of chemical applications and tillage, you're locked into the input cycle. This isn't a flaw in the model. This is the model. This is how you build a market worth hundreds of billions of dollars on a resource that would otherwise regenerate itself. Today, the global fertilizer market is valued at over $200 billion annually, and nearly all of it depends on continuous repurchasing. Nitrogen fertilizers alone account for roughly $60 billion per year, produced through the Haber-Bosch process that consumes approximately 2% of global energy production and generates hundreds of millions of tons of CO2 emissions. Phosphate fertilizers, mined from finite deposits concentrated in just a few countries, represent another $40 billion market, with experts projecting peak phosphorus production within the next few decades. The entire system, from production facilities to distribution networks to application equipment, was built on one assumption: farmers will need to buy these products every single season, forever. Agricultural input companies aren't just selling fertilizer, they're selling a biological dependency that ensures continued revenue. And there's an entire ecosystem built around maintaining that dependency. Research funding flows primarily toward optimizing chemical systems, not biological alternatives. Extension services are often partially funded by input manufacturers, creating institutional incentives to recommend purchased products. Even agricultural education has been shaped by industry grants and partnerships that ensure agronomic students learn chemical-based systems as the default. The scale of this economic architecture makes competing approaches nearly invisible, not because they don't work, but because acknowledging them threatens the foundation of a $200 billion market. In pre-industrial agricultural texts, living soil was simply soil management. There was no other category because there was no alternative. But in modern agronomic literature, it's been linguistically repositioned as biological farming, regenerative agriculture, or alternative soil management, descriptors that frame it as a deviation from standard practice rather than the foundation that standard practice replaced. The original system, the one that sustained agriculture for 10,000 years, has been rebranded as the alternative. Meanwhile, a system that's been dominant for barely 70 years, that requires continuous external inputs, degrades soil structure, and depends on fossil fuel consumption, is called conventional. This isn't just semantics. It's a complete inversion of agricultural reality. Journals publish papers on integrating biological approaches into farming systems, as if soil microbes were a new technology being added to an existing framework, when the truth is exactly backward. Chemical inputs were integrated into biological systems and gradually displaced them. The language shift is profound. Soil health became something you maintain through purchased products rather than through biological stewardship. Fertility became a measure of chemical availability rather than biological capacity. And sustainable agriculture became a niche market category rather than the baseline requirement for long-term food production. The original system didn't disappear. It was rhetorically subordinated, repositioned as a specialty approach for farmers willing to deviate from norms that were themselves only 3 generations old. The infrastructure changed. The research priorities shifted. The economic incentives aligned around annual input purchases. But living soil never actually disappeared. Walk into any forest, any prairie, any ecosystem that hasn't been chemically treated, and you'll find soil microbiomes functioning exactly as they have for millions of years, fixing nitrogen, solubilizing phosphorus, cycling nutrients, building soil structure, all without anyone purchasing inputs or managing the system. Biology didn't need human intervention to work. It needed humans to stop disrupting it. And in pockets of agricultural land across the world, on farms that never fully adopted the chemical system or that deliberately walked away from it, living soil persists, quietly outperforming its industrial replacement on every metric except one. It doesn't generate recurring revenue. These aren't experimental plots or research stations. They're working farms producing commercial yields, building soil carbon, reducing input cost to near zero, and demonstrating that the system agriculture abandoned 70 years ago still functions exactly as it did for the previous 10,000. The microbes are still there, waiting in degraded soils, ready to recolonize and rebuild functionality the moment chemical disruption stops. Perennial systems survive technological revolutions. They survive market transformations. They survive because they're not dependent on anything humans can sell them. Rebuilding living soil is simpler than the industry wants you to believe, and you can start the process today for less than the cost of a single season's fertilizer bill. Begin with a soil test. Not the standard NPK test that only measures chemical availability, but a biological assay that evaluates microbial biomass, fungal to bacterial ratios, and nutrient cycling capacity. Stop tillage immediately because physical disruption destroys fungal networks and kills soil structure. That takes years to rebuild. Plant a diverse cover crop mix that includes legumes for nitrogen fixation, grasses for carbon input, and broadleaf species for deep nutrient cycling, and let those covers grow to full maturity before terminating them to maximize root exudates that feed soil life. If your soil has been chemically managed for decades, inoculate it with commercial microbial products or compost from healthy biological systems to jumpstart populations that may have been Entirely eliminated. Reduce or eliminate synthetic nitrogen applications because excess chemical nitrogen suppresses the biological fixation you're trying to re-establish. In year 1, you're investing in soil recovery, establishing the microbial communities and building the organic matter base. In year 2, biological fertility starts contributing measurably to crop nutrition, allowing you to reduce purchased inputs by 25 to 50%. By year 3 and beyond, AIUI, well-managed living soil system can meet most or all crop nutrient needs without external fertilizer, while simultaneously improving water infiltration, drought resistance, and long-term yield stability. You're not adopting a new technology, you're removing the barriers that prevented the original system from functioning. If this changed how you think about fertilizer, then you're starting to see the deeper pattern. You're not buying plant nutrition, you're buying biological suppression. Every bag of synthetic nitrogen is a purchase that makes your soil less capable of generating nitrogen on its own. Every application of chemical phosphorus degrades the mycorrhizal networks that would otherwise access phosphorus for free. The product isn't solving a fertility problem. It's creating the dependency that makes next year's purchase necessary. And once you understand that, once you see that the system was designed not to feed plants, but to eliminate alternatives that would make the product unnecessary, you can't look at agricultural inputs the same way again. This isn't about being anti-technology or romanticizing pre-industrial farming. It's about recognizing when a technology was adopted not because it was superior, but because it was monetizable. Living soil works. It's worked for thousands of years, and it still works today on every farm that gives it the conditions it needs to function. The question was never whether biological fertility could replace chemical inputs. The question was whether an industry built on chemical dependency could survive if farmers remembered that soil regenerates itself. Subscribe to the Power Roots. We're exhuming the agricultural technologies that were buried not because they failed, but because they succeeded at making farmers independent. The next vault opens soon, and it's going to show you why the most productive food system in human history was deliberately erased from institutional memory in a single generation. You won't find this investigation anywhere else because no one else is asking why the solutions disappeared.